vaccines. So yeah, any questions that you guys have, we will go over them while we're waiting for people to keep filing in. Can we go over vaccines? Yes, we absolutely can. So yeah, the vaccine PowerPoint is very short and pretty easy. I have it pulled up right here, so we can go over that. But it was one that you guys were responsible for looking over on your own. There wasn't a lecture on it just because it's so short, but there's not a whole lot of information on it. So let me pull that up and share my screen. Okay, share screen. Okay. So, all right, shared. And in the chat, are you going to post the Kahoot? Yes, I'm going to post the Kahoot as a link. So I'm going to send out an email after the review is done, just with the Kahoot link and hopefully with the recordings of the videos, but Blackboard is not my friend. So I may send those to Dr. Kokesh to upload, and she will upload those to Blackboard. All right, so vaccinations. Yes, no worries. Yeah, so you guys can practice through that. It's a public Kahoot. You can practice through it as many times as you want. All right, so vaccines. We all know what vaccines are. It is can anyone tell me what kind of immunity it is? What kind of acquired immunity it is? It is active. Is it artificially acquired or natural? Artificially acquired active immunity, yes. So it is artificially acquired active immunity. It is artificially acquired because you are getting injected with the pathogen, kind of a modified version of the pathogen, and you're producing the antibodies yourself. So because your body recognizes that vaccine as other, it makes antibodies itself. So that means it's active immunity and it's artificially acquired. Naturally acquired active immunity is when you actually get sick. And then naturally acquired passive immunity is like through breast milk, like giving IgA is the antibody that is found in secretions in breast milk. And then artificially acquired passive immunity is an injection of preformed antibodies. So vaccinations are artificially acquired active immunity because you're making the antibodies yourself. History, she probably won't super go into history, but still an important guy to know. Edward Jenner is the guy who kind of discovered vaccines. There were already inoculations before that, but he kind of modified the idea. So he was trying to find a way to prevent smallpox. He realized that milkmaids didn't get smallpox, but they had like cowpox lesions on their hands. Cowpox was a similar enough virus to smallpox that the immunity built up by cowpox prevented a person from getting smallpox. He did a very ethically ambiguous, no, it was just ethically bad experiment because it was the 1700s and we didn't have ethics rules. He injected a child with cowpox. The kid like got better and was fine, didn't really get sick because it's cowpox. And then a couple weeks later, injected the child with smallpox, which is a deadly virus. And the child lived, thankfully. But yeah, so Edward Jenner discovered vaccines. That is where VACA, the word comes from, or vaccination. VACA means cow because it originates from cowpox. So yeah, probably won't go super into history, but still good to know. It may be an extra credit or something. All right, so types of vaccines. So there aren't very many. So there's your whole agent vaccines, and there are two kinds of whole agent vaccines, your live attenuated and your killed. You have subunit vaccines, toxoids, conjugated, and nucleic acid. So whole agent vaccines, firstly, the killed bacterium or inactivated virus version. So whole agent vaccines, because it is a inactivated or dead version of the microbe, they're not able to replicate. So it's effective because it's the whole thing, but you may need booster shots because your body mounts an immune response, but it's kind of like, think of it like you're injected with a dead version of the influenza. And your immune system goes, this is bad. And so they start mounting an immune response, start making antibodies, but they also go, It's dead though, so it's not replicating. We don't need to mount like an entire immune response, which is why you would need boosters for any killed or inactivated virus. So yeah, they're like, your immune system's kind of basically like, what's the threat? It's already dead, even after freaking out initially. So yeah, so a couple examples. So yeah, for each kind of vaccine, you should know what it does, what makes it different from other vaccines, and some examples of pathogens that can be prevented by that kind of vaccine. So current whole agent killed vaccines are the rabies vaccine and the injected influenza vaccine. Again, not the nasal spray one, the injected influenza vaccine is a killed whole agent vaccine. Your other kind of whole agent vaccine is the attenuated or weakened bacteria or virus. It is alive, it's just a weakened version of the pathogen. So kind of thinking like if the flu was a dead version of it is injected, there's a version of measles that measles is like arms are missing. It's weakened. It can't really replicate well. It replicates, but it's not virulent. It's not going to cause like a whole lot of disease because a healthy immune system will be strong enough to fight it off. That is why we can't really give whole agent live vaccines to immunocompromised individuals, because the immune system of like a healthy, normal immune system is strong enough to fight off a weakened version of a bacteria or virus. An immunocompromised immune system is not strong enough to fight off the weakened version of this pathogen. And if it doesn't fight it off in time, there's a risk of the weakened version growing strong again and turning back into, mutating back into its dangerous pathogenic form. So that's why we need to vaccinate people around immunocompromised individuals. And then an example of a live attenuated whole agent vaccine is the measles, mumps, rubella vaccine. So measles, mumps, and rubella, in that vaccine, you're getting weakened versions of all three of those things. Any questions on the whole agent vaccines? No. Okay. Oh, yes. One. Nope. Okay. Next one. So yeah, subunit vaccines. Subunit vaccines use a part of a microorganism or virus to best stimulate an immune response. So it's kind of like, a good way to think about it is, you know how vaccines have their protein capsid? It's like just injecting, like you just take the protein capsid without the like genetic material. And you put that in a vaccine and you inject that. And so your immune system still recognizes that's different, that's not us, that's bad. But it's just the like protein coat, like it's just the protein capsid. So if you actually get that virus, your immune system recognizes the protein capsid because it already mounted an immune response against the protein capsid. But this time it is able to fight off like the whole virus. So yeah, safer, it's not the whole microbe, there's less side effects, which is very nice as well. But yeah, it's like injecting just the bacterial capsule or just like parts of a virus. So yeah, it's just part of the microorganism. And then, yeah, so an example, a viral example is the hepatitis B vaccine is a subunit vaccine. And then, yeah, so there's a good example of a virus that's a subunit vaccine, and then bacterial vaccine, Haemophilus influenza type B, or Streptococcus pneumonia is also a good example. So all of those are subunit vaccines. Any questions on subunit vaccines before we get into this slightly more in-depth subunit vaccine slide? Okay, sweet. All right, so conjugated vaccines are a type of subunit vaccine. And like I said earlier, it's like how all squares are rectangles, but not all rectangles are squares. All conjugated vaccines are subunit vaccines, but not all subunit vaccines are conjugated vaccines. Conjugated vaccines are basically more effective subunit vaccines. So it takes a piece of the pathogen and then it ties it to another foreign protein to enhance the immune response. So yeah, developed to get better immune response to polysaccharide vaccines, contain a component of the microbe, not the whole microbe. So again, subunit, it's just a part of the microbe, but it also attaches another foreign protein to just kind of up your immune response. So yeah, basically like your immune system will respond to something that's just a part of a pathogen. But it will respond more to something that's part of a pathogen and also something else, like some other foreign protein. Your immune system goes, that's two whole bad things instead of just one whole bad thing. So it kind of just like ups the immune response. And then examples, again, pneumonia vaccines are examples of conjugated vaccines. Any questions on the difference between subunit and conjugated vaccines? Nope. All right. Toxoids. So toxoids, do you guys remember learning about your bacterial exotoxins? Yep. Yeah. So who can tell me the difference between a bacterial exotoxin versus a bacterial endotoxin? You can unmute yourself or type in the chat. Yes, endotoxins are lipid A. Exo is a protein. Endotoxins are lipids. Very, very good. Yeah. So yes. So endotoxins are lipid A. They're produced by the gram-negative bacteria. This was on your last exam. We don't really need to go over it right now. Still good to know for the final though, so good review. So those are endotoxins. Exotoxins are, they leave the cell. They're produced outside of the cell and they are harder to kill. So it's like an example is like the tetanus bacterium itself is not dangerous. But if the tetanus bacterium is able to produce exotoxins, these proteins, those are what cause the symptoms associated with tetanus. Exotoxins are really bad. They often cause like muscle paralysis. Botox is an example of a bacterial exotoxin, which is why if you inject it in small parts of your face, they stiffen up until your immune system like dissolves it. So toxoids are made from inactivated toxins. Yeah, so they're antigenic, but they're not toxic. Question, are these dangerous factors the endospores of tetanus? So not endospores, yeah. I remember I had that question last semester too. I was like, are exotoxins the same thing as endospores? They are not, they're slightly different. So they're not the endospores are like the little escape capsules, I guess. Like that's how I always thought about them. They're the escape capsules that like when something is in a bad environment, they carry genetic material and protein and they can form into the actual bacterial. Again, yeah, there's the survival mechanism. Exotoxins are different. They're not a survival mechanism. They're just toxins that are produced. They're not like, they're different. Yeah, but they do seem similar. And so, they are fairly, like they seem similar because they're both released. And yeah, again, science, all the things have names that sound similar. You're like endospores, endotoxins, exotoxins, they all sound the same. But yeah, toxoids are made from inactivated toxins so that your body knows how to respond to that toxin. So that's why you have to get your tetanus vaccine every 10 years just to update it. Diphtheria is also diphtheria also creates a toxoid. So DTaP or Tdap vaccine has diphtheria toxoid and tetanus toxoid in it. So yeah, it's a modified it's kind of just like a vaccine, but it just it's the toxin specifically because the bacteria itself isn't dangerous, but the toxin it produces is. So modified version of the toxin is a toxoid, and you inject that and your immune system knows how to respond to the toxin if it actually encounters it then, which is very good and important because tetanus is a horrible way to go. All right, future vaccines. So the main vaccines I feel like you guys will be tested on are these ones, from whole agent vaccines to toxoids. These are the ones that you should like really focus on studying: whole agent vaccines, subunit vaccines, conjugated vaccines, toxoids. There are some future vaccines, and if I'm remembering correctly, these were more just like interesting information to know. Still know them and still study them, like study the last few slides of this, but they're not the most top priority, I guess. Your other kinds of vaccines, like the existing vaccines, those are top priority. Viral vector, the Johnson & Johnson one. I'm not sure. I should know that, but I do not. I do know that AstraZeneca is a viral vector. I don't know if Johnson & Johnson was a viral vector vaccine, but AstraZeneca, the like one used in the UK, is a viral vector COVID vaccine. Yeah, and viral vectors, they're really interesting. It basically uses another virus as a carrier to deliver the like COVID, in this example, antigen into the body. So they remind me of conjugated vaccines. They're not quite the same, but it like kind of mounts an immune response by like getting your immune system to respond to something else, and then the antigen that's like COVID is attached to it. Yeah, so they're cool. And then nucleic acid vaccines, don't know a whole lot about these ones yet. Like they're plasmids with naked DNA injected into muscles with a gene gun. And very futuristic fun vaccines. Proteins simulate humoral and cell-mediated responses. So your B cells and your T cells get involved. Yeah, 2 DNA vaccines or nucleic acid vaccines have been approved for animals. So yay. Cheaper, no need for needles, no need for refrigeration. And so more research, we want more research to go into these, because if we want to mount a vaccination response somewhere that it's hard to have like stable refrigeration, because vaccines need to be kept at a certain temperature, we wouldn't need refrigeration for these. And so trying to do more research into this. And then finally, you have your mRNA vaccines. These are the like Moderna and Pfizer. I'm pretty sure both Moderna and Pfizer are mRNA vaccines, but basically, chat. Yes, thank you for confirming. Yes, so both Moderna and Pfizer are mRNA vaccines. They're really cool. Again, it doesn't alter your cellular DNA. That's what people were so concerned about. They were like, this is altering my genetics. It's not, because it's mRNA. And you guys remember from your like central dogma, it's DNA to RNA to proteins. Because it's mRNA, it's just in the cytoplasm. And it doesn't go into the nucleus of your cells. It floats in the cytoplasm, and then like our RNA recognizes it as a strand of mRNA and says, ooh, you want to make a protein. And the mRNA says, yes, I do want to make a protein. And the protein that the mRNA in the vaccine codes for is a spike protein on a COVID. And so the whole like translation process happens, not transcription, that's DNA to RNA. The translation process happens and makes a protein, and that protein is the spike protein on a COVID. And then that spike protein appears on the cell that has the mRNA strand in it. And your immune system goes, that's not supposed to be there. And then it fights it off. So they're effective. They're very interesting. They, like mRNA vaccines are new, but the technology's not. We've been looking into them for a while now. This was just one of the first ones that we like used it for. But yeah, does this associate with people who thought this vaccine caused autism? I remember Dr. Kokesh having a whole rant about this. So no, it wasn't the mRNA vaccine. A lot of the anti-vax movement, and I will also rant about this. So if you want more information, feel free to ask me later. Andrew Wakefield is on my like Dunzo list. He's public enemy #1 in my heart. But it's not the mRNA vaccine. The anti-vax movement was kind of, it's always been there, but it was formed around vaccines causing autism. And I'm sure when the COVID vaccine came out, there was still like a lot of people yelling about how it probably caused autism. It doesn't. It was specifically the measles, mumps, rubella vaccine, actually, the MMR vaccine. that Andrew Wakefield wrote this paper in the Lancet, which had nothing to do with science, and basically with false science said that vaccines cause autism. But I'm not going to spend the whole review talking about that because that is not going to be on your exam. But if you would like to hear me rant about it, you can ask me later. So that is vaccines. Do we have any questions on those? Yeah. Yeah, I remember the weirdest things from class as well, and not like the main important topics. So if there are no other questions on vaccines, I'm going to stop sharing. What other specific units or topics do you guys have questions on, or do you want to go over? I have all of the slides pulled up. Yes, we can go over phagocytosis. So let me find it. Yeah. Let's go over phagocytosis. So oh, where did it go? It is, do you think Kahoot questions will be similar to the exam? Potentially. So the Kahoot questions, I make them during the lectures, just like what you guys are going over in class. And I send them to Dr. Kokesh, and she uses them sometimes potentially, or she could. I don't get to see the exam or the quizzes before they're made, but she may use them as inspiration for quiz questions or for exam questions. So definitely go over that Kahoot. Practice questions are always good to have. She's used them. Yay! I feel so honored that questions have popped up in the past. So yeah, definitely study the Kahoot. Practice questions are always good. Yeah, it's here. Let me find the, oh, and it's already open to the phagocytosis slide. So here, let me share my screen real quick. Boom, share. All right, so phagocytosis. So I think that this picture is the most useful picture. So it's okay, so I guess there is an adherence. There's an adherence part, but it's chemotaxis and adherence. They're both in step one, like they're both part of step one. Adherence isn't a second step. Ingestion is your second step. So the word adherence comes in the like chemotaxis part of it, like, yeah. So I got a little bit confused there too, with there's adherence and viral replication. I guess chemotaxis and adherence are step one of phagocytosis. Yeah, they're just grouped together. So it's not step one and two. It's both step one. So the way I think about phagocytosis is the first part of it, I like to think about as like, I think of it specifically as like getting into a taxi, mainly so I could remember the word chemotaxis, which has taxis in it. So chemotaxis is the macrophage getting to the location of the pathogen. So the pathogen, like, I mean, it doesn't call for a taxi. It doesn't really want to be picked up. It's more like a police car. So chemotaxis is it getting there. And then step two is ingestion. Chemotaxis and adherence. And then 2 is ingestion. 3 is formation of phagosome. 4, digestion. 5, residual bodies. Yes, exactly. So yeah, it eats it. A phagosome is formed in step 3. And then you remember from learning your organelles, lysosomes are the ones with digestive enzymes in them. So they're your like cell garbage disposals. There's nothing within the phagosome that can kill the microbe itself. So the phagosome needs to fuse with the lysosome. So step 4 is the fusion of the phagosome and the lysosome, which makes a phagolysosome. Very creative. You just smush the two words together. So step 4 is formation of the phagolysosome. And now the digestive enzymes are within the phagolysosome, like where the microbe is. And so step 5 is the cell, digesting the microbe. And then 6, formation of the residual body containing the indigestible material. And step 7 is the discharge. So steps like 1 through 3 are kind of they work with the getting in the taxi thing. Step 4 through 7 is literally just like regular digestion. So you digest it, and then the parts that your body or the cell body can't digest, they get pooped out. So it's like cell pooping. Does this discharge go through the kidneys or? I mean, your kidneys and there's a lot of ways for your body to like get rid of waste products. Most of it is through the urinary system, but you can also like get rid of waste products through sweat and things. So kidneys could be a method that it like leaves. So yeah, it's just floating around in your blood and then it goes to the kidneys and gets like officially discharged from you. So the cell poop and then your like actual body gets rid of it. Hello, welcome. We are going over phagocytosis right now. And yes, so we're just any specific questions that you have, Nicole, just put them in the chat and we will go over them. So are there any other questions on phagocytosis? Mm-hmm. Of course. All right. Let's go over opsonization real quick while we're here. So opsonization, and again, I asked this earlier, but what kind of antibody helps with opsonization? Like which kind of antibody is an opsonin? Yep, IgG. IgG does a lot. IgG antibodies are fantastic. They're the little Y-shaped ones. Opsonization happens because a lot of microbes are slippery and they have methods to get away from being eaten by a cell or being eaten by a macrophage because they don't want to be eaten by a macrophage, they'll die. And so they're slippery or they have capsules that like make it hard for the macrophage to form around them. Opsonins like IgG, they can attach easier to the microbe and kind of form little handles for the macrophage to hold on to and eat. Oh, that was what I was going to go over. I was going to go over the difference between here. Before I move on, so we're on the non-specific host defenses slide. Are there any other questions about the non-specific host defenses? So like primary, secondary responses, because I was going to go over epitopes. Yeah, that's what it's called. So here, I will keep this slide up, but I'm going to open up specific defenses. Can you guys see my screen, like with the specific host defenses? Nope. Okay, I have to reshare it. My bad, guys. Sorry. Stop sharing. All right. So, where'd it go? There it is. Could you see my screen with the non-specific host defenses? I don't think I reshared it. And you might have just been looking at viruses still. I hope not. Okay, that's good. Good. I was a little bit worried there for a second. All right, specific defenses. Sure. Okay, can you guys see it now? Awesome. All right. So this is where'd it go? This. Okay, so epitopes. Because there's a question about this on the Kahoot, and I think this is really something that's easier to see visually. So you have your bacterial cell. It has lots of antigens on it. So these little like pepperoni looking things, little red spots are antigens. Like basically any piece, basically any piece of something that your body recognizes as foreign. It's not going to recognize the whole bacterial cell, but say it has like a flagella. Your body's like, no, we don't like that. That's not one of ours. Say it has like foreign proteins on it. So like the whole things in general are antigens. Epitopes are the specific parts of an antigen that an antibody can bind to. So see how on the antigen, you have like a triangle, a square, and oh no, we only have 10 minutes. Okay, you have a triangle, a square, and a little dome. And then you have antibody B, and antibody B can only attach to the domes. So antibody B recognizes a specific dome epitope. Antibody A is attaching to the triangles. It recognizes specifically the triangle epitope. So epitopes are the specific parts of antigens that antibodies bind to. Antigens are like the whole thing. Does that make sense? It gets a little bit confusing, especially since we like refer to antibodies fight antigens all the time. It's really like a specific part of it. Yes, Brina, what are you confused about? I can try to explain it in a better way. Yeah, think of it as like the head of the antigen. Epitopes are not antibodies. Epitopes are parts of antigens that antibodies can bind to. Yeah, it's the part that's exposed, exactly. Thank you, Megan. Yeah, so it's like specific antibodies are very specific with what they can recognize. And so say antibody B, it doesn't see the rest of the antigen. It only sees these two little domes. And so it binds to the domes, and the domes are the visible part of the antibody, if that makes sense. Yeah, I think describing it as like the head of it or just a specific part of it. Yeah, it's another lock and key moment, exactly. Proteins and the locks and keys, things fit very specifically to other things in the body. Yeah. Oh, that's a good way to think about it too. So yeah, epitope, epi means on top of. Think of it like on the top of an antigen. That is a good mnemonic, Megan. All right. Any other questions about the or about the specific host defenses, or about pathogenicity, anything on viruses, or just questions about study tips? We have about 8 minutes. I would not be opposed to opening up another Zoom if anyone's questions get cut off. But yeah, we'll just see what we can get through in the remaining time. Do you know how many questions the exam will have? I do not know exactly how many questions the exam will have. I believe it'll be probably a similar length to your other exams. Yeah, I don't know the exact number. Brina thinks 60. Yeah. It opens normal time. Yes, it will be opening at the time that the past exams have opened. You can theoretically take it for the whole hour. Like that is your microbiology lecture hour, but I would recommend starting it right at 12 or right as it opens up, normal time around 11:45, just so you can like have time. It is timed, but like just so you can start it, and because it closes automatically at a certain time as well. So start it as early as you can. Yeah. But how are you guys feeling about like your study plans and things? Any like tips for how to study for this one? I would recommend making either a chart or flashcards for, you've been watching the videos. Videos are good and helpful. Yeah, the videos are helpful, and it definitely also helps like to visualize things. And visualization is helpful in this unit, especially like, again, I had to pull up this picture of epitopes versus antigens. Doing flashcards and making slogans. Exactly. Yes. Any kind of mnemonic that works for you. Mnemonics are very helpful, especially in this unit with memorizing steps, I would say. And then again, my biggest tip always, writing things down with the, like physically writing them down with your hand helps for memorization. My biggest tip per usual is you're allowed to have your scrap piece of paper during the exam. It just has to be blank when you hit start. And then as soon as you hit start, before you look at the first question, any mnemonics that you have, write them down, just so you can like see them physically and you're not trying to like pull them back up from your brain in the middle of the exam, because that gets stressful. Made a whole graph for DNA and RNA. Yes, the brain dumping. Brain dumping works. And yeah, I don't know why I didn't use it for all of my exams forever, but it's definitely helpful. Yeah, definitely would recommend making a chart or flashcards, whichever works for you, of the different viruses. What pictures do you think we should look at? I do not know, because again, I don't get to see the exam, so I don't really know what pictures will be on there. Envelope viruses, yeah. So what you need to know about each virus family, what should be on your chart or flashcards, or basically what you need to memorize about each virus family, is if it's positive or negative sense, which again is different than a positive or negative stain, because you don't really stain your viruses, they're not cells. So you need to know the positive or negative sense. You need to know its capsid shape. So if it's icosahedral, complex, or I'm blanking on the last one. That's really bad. Yes, so helical, thank you. Yeah, you guys got this. So you need to know if it is complex, helical, or icosahedral. You need to know if it is positive or negative sense. You need to know if it's a DNA or RNA and if it's double or single stranded. And you need to know if it is enveloped or naked. And then like a fun fact about it is what I call them. Basically, what viruses are in this virus family? What disease does it cause? Like basically the piece of information that you think is probably the most important, like Poxviridae, for instance. Your fun facts could be it's the like family of viruses that has the biggest viruses, or smallpox is part of the Poxviridae family and that's been eradicated. So yeah, you need to know like needing another complementary strand in order to be negative sense. Let me go to the viruses here. I'm going to stop sharing real quick. Sorry. And go to the viruses slide. I think it's viruses part 1. Where did it go? Yeah. Okay. So it is here. It's their polarity. So share screen. Viruses open. Yeah. So single-stranded RNA genomes have a polarity, positive or negative sense. They can, yeah, their nucleic acid can be single-stranded, double-stranded, linear or circular, continuous or segmented. So, and what is a good example of a virus that has a segmented genome, which is why it can like mutate rapidly. Yes, influenza. Yeah, yes, exactly. So negative sense, it's not mRNA. So you have to transcribe it into the complementary strand of mRNA, positive sense to make the proteins. Exactly. So yeah, go over your senses. Yeah, so single-stranded RNA genomes have a polarity of positive or negative. Positive RNA, they can go into the cell and directly function as mRNA. Negative RNA, they must first be transcribed into a positive sense RNA strand, which then serves as the mRNA. So it's like an extra step if it's a negative sense. So yeah, that is good to know, knowing the senses. Got your capsids, your different capsid shapes, different diseases caused by viruses. We have about 1 minute and 42 seconds left. I'm going to stop sharing. So yes, yeah, yep. And flu is in the Orthomyoxybiridae family, yes. So we have 1.5 minutes left. Of course, I am always happy to do a review. I hope it was helpful. And in the last minute, are there any other questions? If not, then I can just kind of like spew study tips. Yay, I'm glad you guys find it helpful. I do enjoy doing them. And also helps boost my own memory about the material. So yeah, make your charts, make your flashcards, go over her study guide, and I would say answer what you can of the study guide in one color without using your notes, and then answer the rest of it that you need your notes for in a different color. So therefore you can like visually see what you like need to work on. Use the brain dump thing. It's helpful, and get a friend to quiz you. Yeah, and motivate yourself by right after this is Thanksgiving break. But yeah, you guys are going to do great. If you have any questions, you can feel free to reach out to me all weekend. But yeah, this is going to cut me off soon. But thank you guys so much for coming, and good luck on your exam. You guys got this. Bye, guys.